First pouch. The first pouch in this diagram, this is the first pouch. It is the purple color, while this is the first cleft. This pouch, it is present between the first and the second arch, lined by the endoderm, while this cleft present between the first and the second arch, lined by the ectoderm. This is the difference. It is present exterior, it is present interior. Now this ventral part is obliterated by the formation of the tongue. Tongue is formed, the ventral part is obliterated, while the dorsal part of this first pouch along with the dorsal part of the second pouch form a diverticulum that is known as the tubotympanic recess. This is an important term. This tubotympanic recess, in future, further development, it has the cranial part and the caudal part. This is the tubotympanic recess. Divide it into the cranial. This is the cranial and this is the caudal. This caudal part, the dilated part, it is the caudal part, dilate and the form. What are the structures formed? It is the middle ear. The middle ear cavity is formed from the caudal part of the tubotympanic recess, along with the mastoid antrum and the mastoid ear cells. These are the three important structures. It is the middle ear cavity, mastoid antrum and the mastoid ear cells are formed from the caudal part, that is the dilated part of this tubotympanic recess. And this tubotympanic recess is formed by the dorsal part of first pouch and the second pouch, while this cranial part, it is the cranial part, this remains narrow. It is not dilated, it remains narrow, and it forms a tube. That tube connects the middle ear cavity with the nasopharynx, that is the eustachian tube. This eustachian tube formed from the cranial part of this tubotympanic recess. Overall, this tubotympanic recess form. What are the structures derived from this tubotympanic recess? It is the middle ear cavity, mastoid antrum, mastoid ear cells, and it is the eustachian or pharyngeal-tympanic tube. These are the structures derived from the tubotympanic recess. In brief, the first cleft and the first pouch, they participate in the formation of the ear. In the formation of the ear, this first cleft and the first pouch participate. The first cleft, it forms the external auditory meatus, while the first pouch, this forms these structures. Now the second pouch. The second pharyngeal pouch, it is present between the second and the third arch, lined by the endoderm. Now, as we discussed earlier, each pouch has the ventral part and the dorsal part. The ventral part of the second pouch proliferate and forms the small buds known as the tonsillar buds. This is the epithelium. This is the second pouch. This is the second arch, and then the third arch. Between the second and third arch, this is the second pouch. Now these epithelium, they proliferate and they form the buds. These are known as the tonsillar buds. And these buds, these are surrounded by the mesenchyme tissue. So these buds invade into the surrounding mesenchyme. And later on, they proliferate and form the tonsil. In short, the second pharyngeal pouch, ventral part, forms the palatine tonsil. The palatine tonsil that is present between the palatoglossal and the palatopharyngeal arches, it is formed from the ventral part of the second pharyngeal pouch. Its epithelium proliferates. This proliferation forms the tonsillar buds. Now they canalized, or we can say the central part undergoes atrophy and they forms the crypts. It is the tonsillar crypts. And this epithelium forms the epithelium of the tonsils. The epithelium of the tonsil, that is the non-keratinized, stratified squamous epithelium. Now in brief, how the palatine tonsil is formed? It has the two sources, endoderm and the mesoderm. Any gland, it is formed by the two sources. Endoderm, this endoderm forms the epithelium of the gland. This endoderm forms the parenchyma, while the mesoderm, it forms the capsules. It provides cytoskeleton of the gland. So any gland, it is formed by the two sources, that is the endoderm and the mesoderm. This endoderm forms the tonsillar buds and then they proliferate tonsillar crypts. These are the buds and they change into the crypts. Now this mesoderm, this is the surrounding tissue. This mesoderm forms the lymphocytes. The tonsil, palatine tonsil, it is a lymphatic organ. So these lymphocytes comes from the differentiation of the mesoderm tissue at the time of the 5th to 6th month of intrauterine life. These lymphocytes are formed. And then this mesoderm forms the stroma. Stroma, that is the connective tissue that provide support of this tonsil. So these two structures are derived from the mesoderm, and as a result of both, it is the palatine tonsil is formed. This mesoderm, it forms the capsules of the tonsil. The capsule of the tonsil, it is derived from the mesoderm. It is a stroma that contains to form the capsule. So it is asked how the palatine tonsil is formed. The palatine tonsils, it is endodermal in origin. It is formed from the ventral part of the second pharyngeal pouch, and its lymphocytes and stroma are derived from the mesoderm, while the dorsal part of this second pouch contribute to formation of the tubotympanic recess. So, in case of the second pharyngeal pouch, the ventral part forms the tonsils, while the dorsal part contribute the formation of the tubotympanic recess.